Class 11, Chapter 12, Respiration in Plants. All of us breathe to live, but the breathing so essential to life. What happens when we breathe? Also, do all living organisms include plants and microbes breathe? If so, how? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know we eat food for energy, but how is this energy taken from food? How is this energy utilized? Do all foods give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms, for their energy requirements, do they eat food? You may wonder at several questions raised above. They may seem to be very disconnected, but in reality, the process of breathing is very much connected to the process of release of energy from food. Let us try to understand and how this happens. All the energy required for the life process is obtained by the oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food. By process of photosynthesis, they trap light energy and convert it into chemical energy that is stored in the bonds of the carbohydrates like glucose, sucrose and starch. We must remember that in green plants too, not all cells, tissue and organs photosynthesis. Only cells containing chloroplast that are most often located in the superficial layers carry out photosynthesis. Hence, even in green plants, all other organs, tissue, and cells that are non green need for oxidation. Hence, food has to be translocated to all non green parts. Animals are heterotrophic, that is, they obtain food from the plants directly, herbivorous, or indirectly, carnivorous. Saprophytes like fungi are dependent on the dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life process comes from the photosynthesis. This chapter deals with the cellular respiration or the mechanism of breakdown of food materials within the cell to release energy and the trapping of this energy for synthesis of ATP. Now, photosynthesis of course takes place within the chloroplast in the eukaryotes, whereas the breakdown of the complex molecules to yield energy takes place in the cytoplasm and in the mitochondria, also only in eukaryotes. The breaking of the carbon-carbon bond of complex compound through oxidation within the cells leading to the release of considerable amount of energy is called respiration. The compounds that are oxidized during this process are known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but protein, fat, and even organic acids can be used as respiratory substrates in some plants under certain conditions. During oxidation within a cell, all the energy contained in respiratory substrate is not released free into the cell or in a single step. It is released in a series of slow step-wise reactions controlled by the enzymes, and it is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be used directly, but is used to synthesize ATP, which is broken down whenever and wherever energy need to be utilized. Hence, ATP act as energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring process of the organisms, and the carbon skeleton produced during respiration is used as precursor of biosynthesis of other molecules in the cells. Do plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require oxygen for respiration to occur. They also give out carbon dioxide. Hence, plants have system in place that ensures the availability of oxygen. Plants, unlike animals, have no specialized organ for gaseous exchange, but they have stomata and lenticles for this purpose. There are several reasons why plants can get along without respiratory organs. First, each plant part takes care of its own gas exchange needs. There is a very little transport of gases from one plant to the another. Second, plants do not present great demand for gas exchange. Root, stem and leaves respire at the rate for lower than the animals do. Only during photosynthesis are large volume of Spire. Gases exchange and each leaf is well adapted to take care of its own needs. During these periods, when cells photosynthesize, availability of oxygen is not a problem in these cells since oxygen is released within the cell. Third, the distance that gases must diffuse even in large bulky plant is not great. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about thick woody stem and roots? In stem, the living cells are organized in thin layers inside and beneath the bark. They also have opening called lenticles. The cells in the interior are dead and provide only mechanical support. Thus, most cells of a plant have at least a part of their surface in contact with air. This is also facilitated by the loose packaging of parenchyma cells in leaves, stems, and roots, which provide an interconnected network of air spaces. The complete combustion of glucose, which produce carbon dioxide and water as end product, yield energy, most of which is given out as heat. If this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell require. The strategy that the plant cell use is to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that energy released can be coupled to ATP synthesis. How this is done is essentially the story of respiration. During the process of respiration, oxygen is utilized and carbon dioxide, water, and energy are released as products. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think of such situation and organisms where oxygen is not available? There are sufficient reason to believe that the first cells on this planet lived in atmosphere that lacked oxygen. Even among present day living organisms, we know of several that are adapted to anaerobic condition. Some of these organisms are facultative anaerobes, while in others are requirement of anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. Glycolysis. The term glycolysis is originated from the Greek word glykos for sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Emden, Otto Markov, and J. Pranas and is often referred to as EMP pathway. In anaerobic organism, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergo partial oxidation to form two molecules of pyruvic acid. In plants, the glucose is derived from the sucrose, which is the end product of the photosynthesis, or from the storage carbohydrates. Sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are photophosphorylated to give rise to glucose 6-phosphate by activity of the enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of the metabolism of the glucose and the fructose are same. The various steps of glycolysis are depicted in figure. In glycolysis, a chain of 10 reactions under the control of different enzyme takes place to produce pyruvate from glucose. While studying the steps of glycolysis, please note the steps at which utilization or synthesis of ATP in case NADH+H+ takes place. ATP is utilized at two steps. First, in the conversion of glucose into glucose 6-phosphate, and second in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde PgAl. We find that there is one step where NADH plus is formed from NAD plus. This is when 3-phosphoglyceraldehyde PgAl is converted into 1,3-bisphosphoglycerate BPGA. Two redox equivalents are removed in the form of two hydrogen atoms from PgAl and transferred to molecule of NAD+. PgAl is oxidized and when with inorganic phosphate to get converted into BPGA, and the conversion of BPGA to 3-phosphoglyceric acid PGA is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP to pyruvic acid. Now question arises, can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? Pyruvic acid is then key product of the glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes. For the complete oxidation of glucose to carbon dioxide and H2O, however, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply. Fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by set of reaction where pyruvic acid is converted to carbon dioxide and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze this reaction. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure. In animal cells, also like muscles during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH+, which is reoxidized to NAD+ in both the process. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose is released and not all of it trapped as high energy bonds of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is the net ATP that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis when one molecule of glucose is fermented to alcohol or lactic acid. Yeast poison themselves to death when the concentration of alcohol reaches about 30%. Now question arises, what then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecules needed for cellular metabolism? In eukaryotes, these steps takes place within the mitochondria and this requires oxygen. Aerobic respiration is the process that leads to a complete oxidation of organic substance in the presence of oxygen and the release of carbon dioxide, water and large amount of energy present in the substrate. This type of respiration is most common in higher organisms. We will look at this process in the next section. Now aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, is transported from the cytoplasm into the mitochondria. The crucial events in aerobic respiration are: first, the complete oxidation of pyruvate by the stepwise removal of all hydrogen atoms, leaving the three molecules of carbon dioxide. Second, the passing of an electron removed as part of hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. What is interesting to note is that first process takes place in the matrix of mitochondria while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of carbohydrates in the cytosol after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by pyruvic dehydrogenase. The reaction catalyzed by pyruvic dehydrogenase requires the participation of several coenzymes including NAD plus and coenzyme A. ना मैं स्टार्ट करे वाला छी. During this process, the two molecules of NADH are produced from the metabolism of two molecules of pyruvic acid produced from one glucose molecule during glycolysis. The acetyl coenzyme A then enters a cyclic pathway, tricarboxylic acid cycle, more commonly called as Krebs cycle after the scientist Hans Krebs, who first elucidated it. The tricarboxylic acid cycle (TCA). The TCA cycle starts with the condensation of acetyl group with oxaloacetic acid and water to yield citric acid. The reaction is catalyzed by the enzyme citrate synthase and a molecule of coenzyme A is released. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation leading to the formation of alpha ketoglutarate acid and then succinyl coenzyme A. In these remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to oxaloacetic acid, allowing the cycle to continue. During the conversion of succinyl coenzyme A is succinic acid, a molecule of GTP is synthesized. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycles where NAD plus is reduced to NADH plus H plus and 1 point where FAD plus is reduced to FADH2. The continued oxidation of acetyl coenzyme A via TCA cycle requires the continued replenishment of the oxaloacetic acid, the first member of the cycle. In the addition, it also requires regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary equation for this phase of respiration may be written as given follow in the figure. Okay. We have till now seen that glucose has been broken down to release carbon dioxide and 8 molecules of NADH plus 2 FADH have been synthesized besides just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither oxygen has come into picture or not the promised large number of ATP has yet been synthesized. Also, what is the role of NADH plus H plus and FADH2 that is synthesized? Let us now understand the role of oxygen in respiration and how ATP is synthesized. ETS and oxidative phosphorylation. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H plus and FADH2. This is accomplished when they are oxidized through the electron transport system and the electrons are passed on to oxygen, resulting in the formation of water. The metabolic pathways through which the electron passes from one carrier to another is called the electron transport system, and it is present in the inner mitochondrial membrane. Electron from NADH produced in the mitochondrial matrix during citric acid cycle and oxidized by NADH dehydrogenase complex 1, and electron are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinone ubiquinol is then oxidized with transfer of electron to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and act as a mobile carrier for the transfer of electron between complex 3 and complex 4. Complex 4 refers to the cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper center. When the electrons pass from one carrier to another via complex 1 to 4 in the electrons transport chain, they are coupled to ATP synthase complex 5 for the production of ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends on the nature of the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of the molecules of FADH produce 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen and the role of oxygen is limited to terminal stage of the process, yet The presence of oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as a final hydrogen acceptor, unlike the photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for the photophosphorylation. In respiration, it is the energy of the oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane link to ATP synthesis is explained by the chemiosmotic hypothesis in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in the synthesis ATP with the help of ATP synthase complex 5. This complex consists of two major components, F1 and F0. The F1 headpiece is a peripheral membrane protein complex and containing the sites for the synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which the proton cross the inner membrane. The passage of proton through the channel is coupled to the catalytic site of the F1 component of the production of ATP. For each ATP produced, 4H plus passes through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. The respiratory balance sheet. It is possible to make calculation of the net gain ATP for the every glucose molecules oxidized, but in reality this can remain only a theoretical exercise. This calculation can be made only on certain assumption that first, there is a sequential orderly pathway functioning with one substrate forming the next and with glycolysis, TCA cycles and ETS pathway following one after another. The NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. Only glucose is being respired. No other alternative substrates are entering in pathway at any of the intermediary stages. But this kind of assumption are not really valid in all living system. All pathways work simultaneously and do not take place in one after another. Substrates enter the pathway and are withdrawn for it as when necessary. ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means, yet it is useful to do the exercise to appreciate the beauty and efficiency of the living system in extraction and storing energy. Hence, there can be net gain ATP of 38 ATP molecules during aerobic respiration of the 1 molecules of the glucose. Now let us compare fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to carbon dioxide and water. In fermentation, there is a net gain of only 2 molecules of ATP for the each molecules of glucose degraded to pyruvic acid, whereas many more molecules of ATP are generated under aerobic condition. NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. Amphibolic pathway. Glucose is the favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for the respiration. Other substrates can also be respired as has been mentioned earlier, but then they do not enter the respiratory pathway at the first step. Figure to see the points of the entry of different substrate in the respiratory pathway. Fats would need to be broken down into glycerol and fatty acid first. If fatty acid were to be respired, they would be first degraded to acetyl coenzyme and enter the pathway. Glycerol would enter the pathway after being converted to PGAL. The proteins would be degraded by protease and individual amino acid after deamination, depending on their structures, would enter the pathway at some stage within the Krebs cycles or even as pyruvate or acetyl coenzyme A. Since respiration involves in the breakdown of substrates, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. But is this the understanding correct? We have discussed above at which points at the respiratory pathway different substrates would enter if they were to be respired and used to drive energy. What is important to recognize and is it is very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrates. Hence, fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as the substrate. When the organism need to synthesize fatty acid, acetyl coenzyme A would be withdrawn from the respiratory pathway for it. Hence, the respiratory pathway comes into picture both during breakdown and the synthesis of fatty acid. Similarly, during breakdown and synthesis of the proteins too, respiratory intermediates form the link. Breaking down process within the living organism is catabolism and the synthesis is anabolism. Because the respiratory pathway is involved in both anabolism and catabolism, it would hence be better to consider the respiratory pathway as an amphibolic pathway rather than as a catabolic one. Respiratory quotient. Let us now look at the another aspect of respiration. As you know, during aerobic respiration, oxygen is consumed and carbon dioxide is released. The ratio of volume of the carbon dioxide evolved to volume of the oxygen consumed in respiration is called respiratory quotient or respiratory ratio. So here is the given formula: respiratory quotient is equal to volume of carbon dioxide evolved by volumes of oxygen consumed. The respiratory quotient depends upon the type of respiratory substrates used during this respiration. When carbohydrates are used as substrate and are completely oxidized, the respiratory quotient will be 1 because equal amounts of carbon dioxide and oxygen are evolved and consumed respectively, as shown in the equation given below. When fatty acids are used in the respiration, the respiratory quotient is less than 1. Calculate for fatty acids, triple methane, and if used as substrate as shown. When proteins are respiratory substrates, the ratio would be about 0.9. What is the important to recognize is that in living organism, respiratory substrates are often more than one. Pure proteins or fats are never used as respiratory substrates. That's it. Thank you.